Imagine being able to watch neurons firing inside a living brain as it hears a sound, makes a decision, or stores a memory. That's exactly what scientists can do using a technique called two-photon calcium imaging. This method allows us to see the brain in action as a story unfolding in light.
Here's how it works. When a neuron fires, it's flooded with calcium ions. Scientists can detect this change in calcium using special fluorescent molecules that glow when they bind to calcium. So now, when the neuron fires, it literally lights up. We can detect this light using a special microscope called a two-photon microscope.
By imaging the changes in fluorescence over time, we can record brain activity, cell by cell, moment by moment, in awake, behaving animals. This technique is revolutionizing how we understand the brain, not just as a map of structures, but as a living, dynamic network.
Here at the Allen Institute, we perform two-photon calcium imaging experiments at an unprecedented scale using a standardized, robust pipeline process. For the Allen Brain Observatory projects, we wanted to understand how neurons in the visual system represent visual information and use this to guide behavior. We surveyed the physiological activity of the mouse visual cortex by imaging the activity of neurons in different cortical areas, different cortical layers, and different genetically defined cell types, while the mice viewed a set of visual stimuli.
Our two-photon calcium imaging pipeline begins with breeding transgenic mice that have a genetically encoded calcium indicator called GCAMP that is expressed in specific populations of neurons. Each mouse undergoes surgery to create a cranial window for imaging, followed by initial mapping of neuron locations, then behavior training, and finally the two-photon calcium imaging itself.
Let's start by visiting the surgical suite, where our pipeline begins.
Welcome to the surgical suite. We have 9 identical workstations. Each of them is equipped with our standard surgical rig and custom surgical tooling that is designed for all of the different kinds of surgeries that we do and allows us to be really consistent with our surgeries across surgeons. For the two-photon calcium imaging experiments, we need to visualize the brain, so we have our surgeons implant a 5mm window into the skull, which allows us to visualize neurons in the cortex. After the surgery, the mice get about 2 to 3 weeks to recover, and then they're passed on to the imaging team.
Once the cranial window has been placed, we create a map of the cortical neurons in that area using a process called intrinsic signal imaging. For the Allen Brain Observatory project, we wanted to map the functional borders of the visual cortex, since the exact shape and location varies between mice. We also needed to map which places in the brain correspond to which points in the retina, and this is called a retinotopic map. The retinotopic map allows us to know which places in the brain will see responses to our visual stimulus. Essentially, we want to make sure that we're recording from the right neurons that are actually going to respond to what the mouse sees in our experiments. This imaging process doesn't use the GCAMP calcium indicator. Instead, we shine a red LED light onto the brain and capture changes in light reflectance caused by different parts of the brain using more or less oxygen, kind of like an fMRI. We show the mouse this drifting checkerboard stimulus to activate different areas of the retina. This allows us to map each of those points to the corresponding area in the brain. Now we know where to record in the brain during our experiments, and the mice can be passed on to the behavior team.
Welcome to behavior. Each of our behavior rooms consists of 12 standardized training rigs, which include a head frame clamp as well as a running wheel that allows the mouse to run freely while being head fixed. The animals will need to be head fixed during imaging to minimize movement of the brain. We perform these experiments in the dark to ensure that the mice are on the reverse light cycle, since mice are naturally more active at night. We use these rigs for habituation, which essentially means that we're getting the animal used to the equipment so that they're not stressed out during the experiment. For the Allen Brain Observatory Visual Coding Project, the mice were simply trained to be comfortably head fixed for extended periods of time while viewing visual stimuli on the screen. Once the mice complete their behavior training, they're passed off to the physiology team for two-photon imaging.
Hi, welcome to our recording suite. This is our two-photon microscope, which uses focused infrared laser to activate the GCAMP calcium indicators that are expressed in our neurons of interest. Our laser pathway starts from back there, that's the laser head. It goes through this, up through the periscope, through the gantry, and then the laser light comes out through this gigantic objective that we have here. Every 12.5 nanoseconds, the laser sends out a pulse of light which gets focused into the brain through the big objective lens. The longer wavelengths of infrared light allow the laser to penetrate deeper into the brain than a conventional fluorescent microscope. This is our behavior stage. It's very similar to the platform that the animals were originally trained on. The running wheel allows the mice to rest or run as much as they choose, preventing boredom and reducing stress during the experiments. We also have all of these cameras. So this is our eye tracking camera, that's our face tracking camera. We have a side view, and then a nose view so that you can see whisker movements. It allows us to extract useful behavior parameters such as pupil diameter, whisker movement, and licking. We use specialized software to control the microscope, the cameras, and other hardware, keeping everything perfectly synced and coordinated. So before starting an experiment, we review the intrinsic signal imaging map collected earlier for that specific animal to guide our targeting to the region of interest. One of our key goals is to record a standardized data set, meaning that we record from the same neurons across multiple days. So we have designed software that helps us return to the same location by comparing the reference image with the live image and adjusting the laser beam to correct imaging depth. Once we're in the right location, we can present the visual stimuli on the monitor and start a recording session. For the Allen Brain Observatory visual coding experiments, each recording session lasted about an hour. We imaged the same group of neurons across 3 days, showing a variety of stimuli, including drifting gratings, flash static gratings, noise stimuli, natural images, and natural movies. And we also recorded a period of spontaneous activity where there was no stimulus on the monitor. These experiments produce a huge amount of data, about 200 gigabytes per session, which is then uploaded to the cloud overnight for processing. We check dozens of quality control metrics on every experiment to ensure proper functioning of the rig, the health and behavior of the mouse, and that the data was acquired properly. And then it's time for data processing.
The data that we get from two-photon imaging is essentially hours of video, showing the activity of hundreds of neurons. We need to process this movie in order to know which neurons are active and when. To do this, we first apply motion correction to the movie, as there's always some natural motion in the brain, even though the mouse is head fixed. Next, we perform image segmentation to define which pixels belong to each individual neuron in the field of view. From there, we can extract the raw fluorescence of each neuron, which we then can normalize to get the change in fluorescence relative to the neuron's baseline fluorescence, and this is something that we call delta F over F. This captures the firing rate of the neuron. Finally, we want to link the activity of these neurons to what the mouse is seeing and doing. So we need to align all of those data streams and package them into a single file for analysis. And for that, we use this pipeline, which will take all of the code that I just showed you and output this final file, which will package the firing rates of every neuron collected for scientists to use for analysis.
Now it's time to share our data with the world. The Allen Brain Observatory visual coding data is available with a graphical interface at observatory.brain-map.org, where you can explore visually evoked responses of individual neurons. And it can also be accessed programmatically for further analysis using the Allen SDK.
We've performed our own analyses of this data and made some cool discoveries, and so have researchers all over the world, including students like you. And there are still so many more questions that can be answered using this data set. We need more people to join in our quest to understand the brain, and you can get started right now using this data set and many others, all freely available at our website.
Happy exploring!